I believe. Thank you.
Yeah. Thank you, Natalie, and thank you, Annie, for the interesting talk. We're going to have Sarah Cox-Vazquez. Are you here?
Hello,
Yeah.
thank you so much for having me.
Hi. Yeah. Okay. So thank you everyone for joining our second keynote talk. For this day, my name is Maimouna Al-Musawa. I'm a PhD student at Center for Tissue Engineering and Regenerative Medicine, the National University, Malaysia. I'm very delighted to moderate this talk on the water-soluble extracellular vesicle probes based on conjugated oligoelectrolytes given by Dr. Sarah. Dr. Sarah Cox-Vazquez is the CEO, and co-founder of Acoerela, which is a startup company commercializing novel dyes for life science. She completed her PhD in 2020 in chemical biology from the University of Michigan. She joined the National University of Singapore, NUS, as a research fellow, where she began working to develop Acoerela's technology and its application in extracellular vesicles. Dr. Sarah, thank you so much for joining us. The floor is yours. Please go ahead and share your screen. I'm going to remind everyone to place your questions in the chat box. We're going to get back to you by the end of the talk, and we'll ask you to unmute yourself and open your camera to have a robust discussion with Dr. Sarah. Yeah. Thank you.
Thank you very much for the kind introduction. Can you see my slides okay? I think that yes. Okay, so, thank you so much to SNEV for inviting me to give this talk. I'm really quite honored that you thought to ask me. So like she mentioned, my name is Sarah. I am currently the co-founder of Acoerela Private Limited, and I'm going to talk to you today about the work that I did when I was a postdoctoral fellow at the National University of Singapore Institute for Functional Intelligent Materials. There we go. So today we're here to talk about EVs and all of their variety. And so for me, what was important when I started thinking about EVs was the different places that I could probe them. So some of the common classic places to probe EVs would be their surface marker proteins or their cargo of nucleic acids or internal proteins. These are very common, and we typically use dyes like, antibody dyes or DNA dyes or protein staining dyes like CFSE, for example. We could think of bioconjugation or molecular beacons to be a few examples. However, there is one kind of universal key factor to EVs that we've been starting to probe, and that's kind of where my talk comes in today. So that's a lipid bilayer. So it's something that's ubiquitous to all EVs. They all have it, and it's this unifying feature that we can easily probe. And coming from a background in biophysical chemistry and being in a lab of materials chemistry, we were thinking of ways that we could develop materials to solve problems relating to the lipid bilayer on EVs. So we're going to talk a little bit about chemistry today. So your typical classic membrane dye chemistry looks something kind of like what we see here. So we have our lipid bilayer, which has our hydrophobic and our hydrophilic portions. Creating this water-loving and water-hating two phases of the lipid bilayer. And our classical dyes like PKH26 or DIR or cell mask, for example, tend to follow this kind of chemical structure that has the cyanine or the dye head group, as we call it, that's sitting on top, kind of followed by some aliphatic tails that kind of go into the lipid bilayer. So this is kind of almost mimicking what an individual lipid looks like per se. And so the problem with that is that if these dyes aren't able to be fully incorporated into a lipid bilayer, they'll actually form their own micelles, just like lipids would. And so the issue with that would arise in they actually will give false positives. And this is due to their lack of water solubility. So this can cause a lot of false positive in your buffer controls, which can lead to kind of a little bit of error in your analysis. So a lot of efforts go into really having to clean up your samples to really make sure that you're getting rid of these false positives if you're using like a classical membrane dye. So these are just a few examples. It's been heavily reported in the literature as well, but some of the techniques that I personally cared about were flow cytometry or DLS, or just even naked eye detection. So these are some data that show just PKH26, which is a very common membrane dye commonly used. So in this example flow cytometry plot from the Beckman-Coulter Cytoflex, where we have the violet side scatter versus the fluorescence intensity. If I didn't tell you that this was only the dye control, you would think that, wow, this is such a beautiful experiment that I've been able to perform. But unfortunately, it's just the dye control. So there's no EVs in this sample. And we can see even by DLS that we've formed some very distinct nanoparticles that are even able to be detected under the naked eye just by shining a laser pointer through a solution. And so this can really cause some problems in your analysis because they really do look just like EVs. So like I mentioned, we come from a materials chemistry background. I'm not a classical EV person. My background is in biophysics. So, the lab that I was working in, the Bazan lab at NUS, we have a series of materials called conjugated oligoelectrolytes. And so they're a water-soluble material that has some very interesting properties. And so, we have been developing these probes in the lab for quite some time, actually. And it wasn't until we encountered our friend, Basie, who told us about EVs, actually. She was a rep at Beckman, a salesperson at Beckman-Coulter at the time, and she knew that there was really a problem in detection of extracellular vesicles on flow cytometry. So she told us, why don't you give a try to your materials and see how they work? And luckily for us, they tend to work very nicely for detecting EVs on flow cytometry. And so it brought us to design a new palette of these types of materials, which we published earlier this year in January 2023 in Science Advances, where we developed a series of different colors and applied them to detection of a variety of methods of EVs on fluorescence-based techniques. So let me just tell you a little bit about COEs, or conjugated oligoelectrolyte is the full name. So it's actually a water-soluble small molecule, and that's quite unique to COEs, but they actually don't mimic the whole domains of lipid bilayer. Like I mentioned before, they have the hydrophobic core with the hydrophilic tails. And the really nice thing about being a synthetic chemistry group is that we can really easily tailor our design. So they're kind of like building blocks, or like Lego blocks, where you can kind of put in the qualities that you want. And in our case, we were really interested in changing the color. So that's where changing this hydrophobic core comes in. So you can kind of imagine it like we could put in different color Legos into the core of the COE to change its colors. So down here in this picture is from our paper, where we show 7 different colors seven different unique colors that we were able to achieve just by changing this inner conjugated core. So this is the structures that are in the paper, and then a few of them have been commercialized by Acoerela as well. So just a bit more background on the kind of photophysical properties before we really get into the dyes. So you can see just by taking a look at that very top row that we do have a variety of excitation and emission sources. So, they can be easily excited on any laser, any system that you're using. And that was kind of really our driving force, because at the time when we started, we just had one color, but we wanted to be able to develop something that a lot of researchers could use, depending on their instrument or what their needs were, so that they could easily multiplex with other dyes. And they do have a very high quantum yield, and they are what we call fluorogenic, meaning that they only light up when they find a lipid bilayer target. And we do know that they're also quite non-toxic in terms of cell-based assays. So if we were to take a look at how the COE-Dye might compare in terms of a cross-section to a classic lipid dye. Like I mentioned earlier, the classical dyes tend to just kind of sit on top of the lipid bilayer because they mimic 1/2. Whereas COE is actually mimicking both domains of the lipid bilayer, the hydrophobic and the hydrophilic core. So it'll actually intercalate all the way from the inner to the outer leaflet, spanning the entire lipid bilayer. And this is a very unique feature of these dyes. It's the first type of dye on the market. And yes, so this is an example of this light up mechanism that I mentioned. So if we, just in this small video that we took actually on an iPhone, holding it under a UV light, in one vial we have the COE just in water, and the other half we have it in a really concentrated solution of lipid bilayer. And you can see just when you hold it under a UV light that it really does exhibit kind of this light up property or this light up mechanism to really see the kind of effect or the fluorogenic nature of the dye. So if we were to compare labeling for EVs, we kind of drew up this schematic and why something like this would be important. So if you think about, you had your EVs and your COEs in solution, if you were to mix them together, you want something that's going to be responsive to, you want something that's going to be responsive to to whatever analyte you're looking at, in this case, the lipid bilayer. So the cyanine dyes also will show some response. However, they're going to produce a lot of these kind of aggregates. And without being able to remove them, it's going to be quite difficult on something like flow cytometry, for example. So on the top, we again just have the dye controls. So both on the Cytoflex and on the NanoFCM NanoAnalyzer, you can see just a comparison between kind of comparable dyes, our COE 600 versus PKH 26. You can see just on the left-hand side of the screen, again, this is just dye only. There's no EVs that are in this sample. But when we look at PKH 26, on both of the instruments, we see a very, distinctive population coming. This could be a bit confounding for researchers. So this is again due to the water-soluble nature of the dye. So they are not going to form these kind of aggregates or false positives in your buffer, as well as this kind of fluorogenic nature. So the fact that they actually turn on when they find a lipid bilayer. So a few of the photophysical properties again. So we wanted to kind of show visually really that the dyes are water soluble and they're very easy to clean up. So we've performed a few experiments using Amicon filters. So I can direct you to the left-hand side of the screen where we have two colors, so COE-BO and DIR. In the top half an Amicon. So this is the Amicon filter that has a 100 kilodalton cutoff. And so you can see that there's a very colored solution on top. Let me just switch to laser pointer for you. You can see that there's a colored solution on top of the Amicon. So this is before putting it into the centrifuge to try and filter it through. So after following the manufacturer's recommended directions, we filtered it through the Amicon, and we can see for the COE-BO here that the color goes all the way from the top of it and passes easily through the filter. However, with something like DIR, because it's forming these nanoparticles, it really does get stuck in the filter. So you lose all of the color, so all of the dye actually getting stuck in the filter because they're quite large. That's why on the bottom, the solution is very clear. And so we did demonstrate this by measuring the before and after two of the absorbance of the solutions that are in either the top in the blue line or after filtration in the red line. So you can see that before and after for COE matches perfectly because the dye is water soluble, it can easily pass through. And we do know that it's only emissive in the presence of lipid bilayer. So we've tested it with highly proteinaceous solutions like FBS or BSA, and we don't see any emission happening. So in this fluorescence intensity plot that we have over here on the right, you can see the COE and PBS is in black, so very small line at the bottom, and FBS is in red. So this highly proteinaceous solution that's overabundant with protein is not going to induce the emission of the COE. However, when we add it to something like small ulamellar vesicles, which is a good model system for EVs in terms of photophysical and biophysical experiments, we can see that the emission intensity really does increase and does take off. We also know that it doesn't change or damage or swell the lipid bilayer. So because it's mimicking the whole domain of the lipid bilayer, it's quite compatible, biocompatible in that sense. So it doesn't actually change the size of liposomes or exosomes, for example. So here we have in black just SUVs before staining, and then in red SUVs after staining with COE. We can see a very similar DLS profile. But, you know, seeing is believing, and we wanted to make sure that we had some cryo-TEM evidence to also kind of back this up. So we performed a few experiments as well on the cryo-EM, looking at just control liposomes as well as control EVs before and after staining with dye. And we can see that, you know, we still see a very intact lipid bilayer. We didn't see any evidence of damaged EVs as well. And we also were able to look at this on flow cytometry. So the NanoAnalyzer is really great for these types of experiments where you want to know size and concentration exactly. So we were able to look at some RBC EVs before and after staining. And we can see that the profile, the size distribution profile, as well as the concentration remained unchanged. So we got a size of 116 nanometers plus or minus 20 before or after staining. So this is again data from the NanoFCM NanoAnalyzer where we have the size versus the fluorescence intensity, just to show this we're not changing the size. So the COE EV staining protocol for flow cytometry, I know I gave you a little bit before, but let's talk a little bit about the protocol. So traditionally, if you were going to stain with perhaps antibody dye or another type of dye like PKH26, you probably would need some washing or purification steps to get rid of anything that's not bound. And, you know, in some cases, you might also need to do that with COE as well. But, you know, because of its fluorogenic nature and its water solubility, we are able to kind of perform a no wash, very simple protocol. You can simply incubate your kind of concentrated, your purified EVs, your enriched EV sample, if you will, with the dye, incubate at room temperature 37 for about 30 minutes to 1 hour. And then the dye will spontaneously intercalate into the lipid bilayer. And so if we want to take a look at some flow cytometry plots for that, on the bottom half of the screen, we have just our dye controls, just our PBS controls. And these are 3 different color examples that we currently have. So even on PBS with the violet side scatter and the fluorescence figure, so here we're looking at violet 525, for example. We still have some kind of scattering that's happened. So something we know is passing in front of the laser. And if you're not super familiar with flow cytometry, each dot that you see on one of these types of plots, so this is kind of like a heat map plot, each dot that you see represents an event. So it's something that has passed in front of the laser and has triggered detection. So even with something like PBS, which should be extremely clean, we still see some of these events passing in front of the laser. So we still have some kind of background that you're working with. So even just for the COEs and buffer, we see that we have some background that's coming out that's just from the background of the noise and the background of the laser. So if we take a look at the exosome sample, so these are PC3 exosome standards from ABCAM at a concentration of 1 × 10⁶ or 1 to 1 × 10⁸ particles per mil. So we can see that we have a lot more density coming up. So our plot is now a lot more dense. We do know something is passing in front of the laser, but we don't really know what the composition. If we're just looking at side scatter, we're kind of losing out on some of the beauty of flow cytometry, not using that fluorescence capability to kind of distinguish our samples. So if we were to add in our dye, we see that here on the top, so we add in the COE 430, 490, and 600 in different concentrations depending on the intensity of the dye. We can see that the fluorescence intensity really helps to kind of pull these things out of the noise, to say. So it helps you to really identify in this population what your background is and what your actual stained population is. And so we do know that the dyes are quite stable, actually, once they're intercalated into a lipid bilayer. So we did hear when we were kind of talking to people about this problem that this dye exchange tended to be something that was an issue for them. So we wanted to see like how the COE dyes would actually interact in this problem. So we took two different populations and stained them individually with COE. And we allowed this to incubate on the bench top. So for 1 hour, just at room temperature, sitting covered with under foil in the room. And we can see that we have two very distinctive populations, both on our one fluorescence intensity channel, so our red channel versus our green channel. And we don't see any kind of co-stained population, so we didn't see any dyes mixing. So we decided to leave that again on the bench for 24 hours, and our results were almost identical. We didn't see any of this interparticle dye exchange, so the dyes weren't going from one population of green to red, and they weren't kind of intermixing onto the same particle. And just to prove our point, we also performed this experiment with DIO and DID, some of these kind of classical membrane dyes. And just after 1 hour, we saw a whole bunch of positive signals in this kind of dual stained quadrant, where we did see a lot of the dyes going back and forth between the populations just after 1 hour of mixing at room temperature. And so I think kind of one of the things and one of the beauties of flow cytometry specifically is actually being able to look at multi-parameters. So being able to see multiple populations in the same sample just using dyes and colors. So antibody co-staining was one thing that we knew was going to be really important, and it's been quite important in the field. And honestly, one of the reasons why we started working is because we knew that antibody staining wasn't giving kind of enough to researchers, and that's why people were getting into membrane dyes. And because we know that the COEs don't light up in the presence of proteins, we wanted to develop kind of this antibody co-staining assay. So we did learn that the COE dye is compatible with antibody co-staining in a no wash or no purification type of assay. So you can incubate your antibody with the exosome for about 1 hour at room temperature, just pipetting to mix to make sure that antibody gets bound. And then you can take that sample and analyze it for flow cytometry and kind of see what your background populations are. Then you can add the COE and then incubate as previous, and then analyze that population again by flow cytometry. So if we were to see just kind of our first step, so binding our antibody onto the EV. So these again are PC3 exosome standards, which we purchased from Abcam with BioLegend antibodies that are conjugated with PE. So we have kind of the classical palette of antibodies, the CD9, the CD63, and the CD81, as well as a cocktail of all of them. We can see that they give different levels of positive signal with, this is flow cytometry from the new BD machine. And at the bottom, we have just PE is our x-axis versus COE 490 will be our y-axis. And we can see that we have some positive events, but we are still quite overwhelmed by the noise of the system. So ranging all the way from about 17 for our highest markers, so CD9, down to about 29% for our cocktail, and as low as 4% for CD81. And so the nice thing about kind of adding in this second parameter, and the beauty of flow cytometry is getting this multi-parameter kind of data, is that, you know, now we can really see more of what our total population is. So if we were to add in the COE 490, we see that we have a very high positive of about 56%. for our COE-490 staining. And then when we add that in, we still see the same breakdowns, the same ratios for our antibody stain, which we can see over in this bar graph on this side. But now we're actually able to see kind of more of what our total population is. So we can use this as a gating strategy to be able to identify kind of what is your total population, and then what is the percentage of that population that is actually carrying an antibody, for example. And we didn't see any kind of off staining effects. So nothing that was introducing any type of artifacts with the antibody staining, which was exciting to us, being able to see these same breakdown in percentages. So the water solubility, like I mentioned earlier in that experiment where we performed with Amicon, just flowing the dye through the filter, we wanted to see kind of what is the actual downstream applications. Like, yeah, it's really great that we can pass the dye through the filter, but what would that actually mean for someone that would actually be using it? So we learned a lot about the co-culturing experiments that people are quite interested in. So they want to be able to stain their EVs and then see how they interact with cells. So we worked on developing those assays in just an Amicon ultrafiltration, where you can just start with your stained EVs and then add them into the Amicon, filter to spin any of the excess dye, and then recover your stained EVs. So we started just with the dye controls, again, in kind of like our previous experiments where we showed just the COE versus the DID control. So because we do know that in this experiment with just the dye control, we're going to lose all the dye because nothing is going to be bound to anything for the COE because it's water soluble. So we should have a very clean background just for those cells. So this is data that we took from the Amnis image stream, which is a very, very cool instrument. If you're not familiar, it's a little bit like if fluorescence microscope and confocal kind of had a baby. It's kind of like a really cool hybrid between the two, where it actually allows you to look at individual cells and the fluorescence and the distribution of that. So for the COE, we don't see any kind of fluorescence happening. However, for the DID, we kind of see these plotches just for the dye control, again, kind of all over the cell, but we don't have any EVs here. So again, kind of showing that false positive need for kind of these more time, labor, and intensive cleanup methods for the traditional type of dyes. So we, again, incubated the EVs and the DID labeled EVs with both of these dyes at the same concentrations and performed that kind of cleanup with the Amicon. And then we wanted to see how these labeled EVs looked over time. So we incubated them at different time points at either 2, 4, and 8 hours, allowing them to interact with the cells more. And so on the COE side, we can see that as time goes on, we start to see more and more fluorescence coming from the cell. However, for the DID labeled EVs, we see kind of a lot of more of these blotches, and they tend to be more kind of different from how the COE looked, where it's much more punctal-like and a lot more splotchy and less easily resolved. And so we were able to quantify this. We analyzed over 1,000 cells in each experiment and on the Amnis image stream. And we could see both the kinetics, so the percentage of labeled cells, so how many spots they were actually getting over time, and versus how many actual spots were in the overall cells. After about 8 hours, we did see on average 15 different spots on both samples, plus or minus 10. So there was quite a lot of variability in the amount of spots that we were seeing, fluorescent spots. So with that, I would like to thank a lot of people that helped with this work. A lot of people in the Bazan lab, as well as a lot of people in the MD1 flow unit that really helped me to learn flow cytometry. Like I mentioned, I'm more of a biophysical chemist by training. So I did have a lot to learn to get into the EV field, and luckily we had a lot of experts as well as a lot of great collaborators when I was at NUS. And so I do have a quick announcement. So I do know that we have a lot of students here, and we are really interested at Acoerela to kind of help you kind of get out and share your work. So we do have a travel grant that we're currently circulating for people to use, to travel to conferences and share their work. So if you're interested, you can fill out our travel grant application and we'll get back with you. And with that, I'm happy to take any questions that you might have. Thank you so much for the opportunity.
Thank you so much. And that's so generous of you, to provide this travel grant. Thank you so much for the interesting talk. I learned a lot, and it's quite interesting because it solves many problems for the existing labeling techniques that we use to track and label EVs. So I would like to open the floor for questions. If anyone has a question. Yasmin, you want to unmute yourself to ask your question? Okay, or you want me to read for you? Okay, so Yasmin is asking, what is the waiting time for dye control in comparing to two techniques in fluorescence microscope?
Oh, okay. So I'll just go with this experiment. So for the dye control, we waited before doing the purification about 2 hours. So we followed the shortest incubation time to see the uptake for that. In terms of kind of your flow cytometry or other fluorescence-based assay, you can incubate anywhere from 30 minutes at room temperature up to overnight if you wanted to in the fridge. It's really kind of depends on what your kind of research question is and how you want to use the dyes and what your applications are. But there's kind of no set time. I wouldn't say incubate for a week because then kind of what could be happening to your samples. But it's quite flexible.
Yeah, wonderful. So I might have a question. So have you tried this in an in vivo system to track EVs after administered? To study the biodistribution of EVs, for example?
Yes. So we do have a nice collaborator who just told me actually that their publication was accepted. So I personally haven't tried it for in vivo work, but we do have a collaborator who has used it, who says she's getting ready to publish, tracking some of her EVs in vivo. So exciting for both us and them.
Because I'm interested to know how about the stability, the photo stability inside, and the specificity as well. So Agatama, you want to ask a question?
Hi, Sarah. First of all, I really loved your talk. It's very interesting actually to know and explore the concept of water-soluble dyes. I just wanted to ask you whether you can summarize the differences that lies with using these dyes as compared to pKa26, because that's one of the most common ones that we do use. So what are the points you could just say would be better if we use this instead of the normal pKa?
Sure. Water soluble, so it's pretty easy to use in your downstream applications, as well as its kind of light up nature. So it doesn't start emitting until it finds a lipid bilayer, an EV in our case, and very low false positives in your controls. So very easy cleanup. You don't have to do ultracentrifugation like with a density to get rid of kind of those dye aggregates. Ease of labeling would be kind of the quick ones.
So would this be possible that, just like try to think it out loud, that instead of characterizing EVs, if we just use this dye, and if you want to eliminate some other technique and only if you want to go for characterizing EVs using this particular dye, can that be something that we can take up? Is it ready in that level to be used as a characterization tool?
A scattering machine tool?
Yeah. Like basically, like for instance, with EVs, we do our isolation, and then we go for maybe Western blot or something to identify that what we've got is definitely an EV population. So instead of just going for those, can we just use this instead? Maybe we can have like another backups to prove that, what the populations that we've got, and they're definitely EVs, but can this be used to replace any of the age-old techniques that we're using?
I think a replacement is kind of a stretch if you're just thinking about a dye. You kind of need to apply it in kind of a known sample, I would say. You can certainly use it as something as kind of a lipid bilayer kind of determiner, whether or not you have something that's a lipid bilayer. And then I think you have to get into kind of what is an EV, what is kind of like what are you specifically interested in learning about your EVs? I guess if you're only interested about learning whether or not you have a lipid bilayer in your sample, then I think, yeah, I think it would be possible to develop an assay. But I think it's, when you're looking at EVs, you need to know something about the size as well as the concentration, the particle count, any other markers that you're interested in. So I think it fits nicely in kind of to a palette of different experiments that you can think about for analyzing your EV sample, but I wouldn't take it as the sole source for identifying whether or not you had EVs.
All right, thank you.
Yeah, so I have a follow-up question. Sorry, Natalie, for keeping you waiting. Yeah, so about the specificity of this dye, because it's a lipophilic. So if you have, for example, lipoproteins in your preparation, or you're studying a complex biofluid derived EVs, so how does this perform?
Yeah, so let me just go quickly to kind of one of these images, for example. Oh, is the one I want? There we go. Oh, this one. Okay, so if we think, it depends on the lipoprotein, right? I think lipoproteins are so broad, just kind of like EVs are so broad. You know, we have these very small, dense lipoproteins that are kind of more micelle-like, versus kind of like these big, large VLDLs, which can, you know, looks very much like an EV. So the thing with lipoproteins is that they're different from EVs in the fact that they usually only have a lipid monolayer that is surrounding a very dense cholesterol core. And the thing with these dyes is they actually span the full lipid bilayer in order to become emittive, to become bright. So if you look at something that has like a lipoprotein, you can see that, you know, you might get a little bit of background fluorescence, some residual fluorescence, but the fluorescence intensity is going to be much higher when you have something like a lipid bilayer over a lipid monolayer, I would say. And I don't want to be kind of give like a blanket statement that yes, it's going to be, you know, this big distinguisher between a bilayer versus a monolayer, but you will see some differences in the fluorescence intensity and almost kind of the properties of the dye when it's interacting with kind of these two different types of lipid samples.
Yeah, how about leaching out, for example, from the EVs and binding to cells when you inject them in vivo, for example? Have you tested this possibility?
Yeah, so we do know that they are like very stable once they bind. Once they find a lipid bilayer, they really kind of don't like to leave. Well, I think I skipped the slide. They really don't like to leave. They don't really leach out. So we do know that the binding is quite stable. The staining is quite stable. It doesn't really kind of leach out. So as long as your sample is stable, so your EVs are stable, the dye will remain.
Thanks. Yeah, Natalie, you want to ask your question?
Sure. No. Thanks, Sarah, for a great talk as always. Maimona, you kind of stole the question I had about the complex biofluids and how the dyes perform in the presence of all these other like non-EV particles. But then I started to think as well, how much work has been done with the dyes on like not your small EVs, but large EVs, for instance. And also, like, I gather these are all like mammalian EVs that you've been working with. Do you know if there's any differences if they're used for, say, like non-mammalian cell-derived EVs, like now plant EVs are a thing, and bacterial EVs, like has that been done yet?
So it depends on the bacteria. So these stain really nicely for gram positive and not gram negative. And so it depends on the type of bacteria. Actually, before we started working on EVs, we were actually working on bacteria. So we do know that they stain bacteria really nicely. They stain mammalian cells also really nicely. But as well as food-derived EVs, we do have some people who work with us that have tried a variety of different types of foods. So, you know, all the way from milk to blueberries and ginger, I know, are some of the things that they've worked with. So they do work for that. That was one part of your question. There was another part of your question.
Yeah. So the just before we move on, so what about yeast EVs? Has anyone done yeast EVs?
Anyone done yeast EVs? Not to my immediate knowledge,
Okay,
but you know, people we have started selling, people do buy, and I don't necessarily ask what they're going to use it for. You know, I let people do the research as they want.
Of course. So the other part was just the size of the EVs. Does that make a difference? I mean, they're all lipid bilayer, so I would imagine not, but I know that you did functional studies with them and it didn't seem to affect the uptake of the EVs if they had incorporated the dye or not. So the probes, sorry. So yeah. Do you have any comment on the different sizes, or you haven't noticed anything different?
For me personally, I've only worked with a few different types of EVs. So like the RBC EVs, the red blood cell EVs, are actually quite large for an EV. You know, they tend to be, depending on which technique you're using, you know, 115 or 200 nanometers. They're much larger EVs. We do see really nice staining with those, even down to kind of like the smaller size EVs that are about, you know, 70 or 60. Kind of more like a microvesicle. Personally, I haven't tested it, but I would imagine that they stain equally as nice because they are, again, a lipid bilayer. You know, this is a lipid bilayer dye applied for EVs.
Yep, excellent. Thank you. Thanks.
Yeah, thanks, Natalie. Thanks for all the answers. Yeah, so I think we're coming to the end of our session. Thank you so much, Sarah, for that informative.
Thank you so much.
Wish you all the best. Yeah. So I'm going to pass the floor